Hello, welcome back to biology. You recall last time we looked at functions of organelles, so this is going to be a continuation of that. When I said last time, of course, referring to biology 3, because the last one was the recap. So, yeah, functions of organelles continued. Okay, so let's see what we got here then. All right, so the next thing we're going to speak about is the ribosome. And you recall the structure of this was, we'll just use maybe this colour here. That lilac maybe? I guess the coloration is not so important because, in terms of how these things look in reality there. And I will be putting some more pictures here. So basically this is a more, a little bit more accurate representation of a ribosome. And I'll just put over here, we've got these small subunit. And this is the large subunit. Okay. Alrighty. Um, okay, so what is this then? Well, it's a very small organelle. Um, um, that is either going to either floats free in the cytoplasm. One of these days, I'll look at these settings, try and get everything looking clearer in this. Sometimes when I do try and cross the eye, dot the i's and cross the t's, it doesn't quite work out. But oh well. A very small organelle that either floats free in the cytoplasm, or is attached to the rough endoplasmic reticulum. Call this two. It's the rough and the smooth, RER and SER endoplasmic reticulum. How did I think about doing? I think I may have thought about doing this. The function. So just put function. The site where proteins are made. And obviously proteins are going to be very, very important. As we move ahead. And yeah, I'm probably gonna focus on a lot of this cell stuff for a while. I think it's a good idea. Before we get into other topics, because it's such a, so yeah, there's the ribosome then, ribosome. Okay, let's do another. And these are mainly, well, so far anyway, they've all been organelles that have been in the pictures drawn in 1 and 2, and also in some of those images that I found in Biology 4. So I want to try and get this nice sense of continuity, sense of recap always, now and going forward, just to, yeah. So let's do the rough endoplasmic reticulum. Again, I'm going to use abbreviations where possible, I think. Because it just seems like the right thing to do. Yeah, actually, I might do if I did a thinner line, maybe I could do that black and then this yellow. Maybe that would be better. So yeah, so it may sometimes be so when we do the rubber independent reticulum, we obviously have to keep in mind the ribosome might be attached. Yeah, well, in fact, the surface of it is going to be covered with them. And this is to do with protein folding, which is a topic that also comes up very, very often. So yeah, what's the best way to do this? I wonder what this line is actually going to look like. It doesn't actually give me a fill color. That's the strange thing. But I actually kind of prefer it. It kind of is. It looks a bit better than to do it like this, because you actually get something which perhaps is approaching something that I might actually draw. So we'll do something like this, and then yeah, it's just like, well, that just looks like an H, but yeah. So we could just have all these rough. Well, I should probably make them a bit more circular actually, because these are actually ribosomes here. So let's go back to a red. Let's go back to a bigger thing. Okay, so we're going to have. So this is pointing directly to here, really. So it's a ribosome. So we've got the proteins made in the ribosome there, right? This is just like the fluid. And I could almost actually just put it on the side. I may as well mix things up a bit. So we could say it's a system. So that's always a good word to use. A membrane system. And it's like containing a volume filled with fluid. Okay. So obviously, on the surface is yeah, covered with ribosomes. So that's fairly clear. I do like that drawing actually more. It looks a little bit more how I'd like it. So the surface is covered with ribosomes. Function. So the function is protein folding and processing. So we've got the ribosomes making the proteins, and the rough endoplasmic reticulum is folding and processing them. Folding, processing from proteins engineered at Ribosomes. Okay, good. So yeah, that's the rough endoplasmic reticulum. How many did I do before? I did 4 last time, so I could do 4 here. Well, there's no actual limit on how many I can do. We'll see how many I get through anyway. So that's the rough endoplasmic reticulum. And now we've got the smooth endoplasmic reticulum. So as you can imagine, the difference being between the rough and the smooth is that there's no ribosomes. That's why, and that's the rough. Ribosomes are rough. There we go. That's one way to put it. What are we looking at here? If I do, let's do go back. Yeah. Okay. What colour is this going to be? Black. Yeah. So yeah. No, can I just fill that? Well, I didn't fill the last one. It's quite hard to fill a structure like this, I would imagine. Let's try it though. Oh no, it's alright. Is that going to let me fill it? Okay, so it's quite accurate actually. I'm going back to the pencil. Actually, no, I wasn't using the pencils, I was using a brush. Oh no, I've changed it. So just like that. And do fill with yellow back. Oh shit. That doesn't actually look too bad though. Huh. That does not look too bad. I'm tempted to actually change the colour, because I noticed in the thumbnail I don't really like having the white background. See the, oh yeah, it's funny. It's funny how that's actually gone, that's happened. But anyway, yeah, let's just, what are we looking at if we do that? Yeah. Alright, so these are basically just, long sort of like, just thin structures that are smooth. That was not the best one. Yeah. So I'll just put here. I'm gonna try. I wonder. This might be alright. I know because I'm well aware that I need to improve like how all this stuff looks. No, that's not right. Similar to Aria. But with no ribosomes. Okay. The function of this is something I haven't actually spoken about yet, so I'm just going to write the function here, and then we'll speak about this in a moment. The function is yeah, it does look much nicer actually like this. Synthesizes. Yeah, it was good for the way I do the S. I wonder if that's. I think I might even have a name the way I do the S. Can't do double S like that though. Synthesis processes lipids. Okay. Okay, so I think actually I'm going to leave it there because what the next up we're going to have, and I think the next episode